Okay. So is my screen visible to you?
No, ma'am.
No. 1 minute. Yes. So here I'm going to share my screen. So,
Yes,
okay.
ma'am.
Okay. See. Then we discussed in the previous class, we discussed how the 5 kingdoms are divided. That means the Whittaker classification. Okay.
Okay.
So basically, who else are classified? The living organisms are classified by many people. Okay, what are the popular classifications? There is 2 kingdom classification and 5 kingdom classification as well. So Carolus Linnaeus is the person who classified organisms into 2 kingdoms. Okay,
Yes, ma'am.
what are the 2 kingdoms? Kingdom Plantae and Kingdom Animalia.
Animal.
Okay, so what are the demerits? So did the prokaryotes gets the proper place in the 2 kingdom classification? No. Okay. Prokaryotes which are not having true cell, true nucleus, that kind of organisms are not, neither considered as plants nor as animals. So these are not getting the proper place in the 2 kingdom classification, because there are only 2 kingdoms, plants and animals, and these prokaryotes which are not belongs to any one of them are not properly classified. So that is one of the demerit. And unicellular, there is no difference. All unicellulars are also comes under one same, same kingdom, and all multicellular organisms are not differentiated. Both are both are placed together. Okay, so that is what the Linnaeus classification is. So next comes the 5 kingdom classification that was proposed by R.H. Whittaker. Okay, in the year of 1969. All are important. You need to remember all these things. So what are the 5 kingdoms? Monera, Protista, Fungi, Plantae, and Animalia. Okay, so basically, I think we completed Monera in the previous class, which is completely consist of prokaryotes. That means there is no nucleus. The metabolism is either autotrophic or heterotrophic, and there is no presence of nuclear membrane and cell organelles. Okay, membrane bound organelles and nucleus is absent in monerans. Okay, so basically, kingdom Monera consists of bacteria. So they are only visible through microscope, as those are very small in size. Okay, they show extreme metabolic diversity as well. So the metabolic system from one bacteria might be different from the other bacteria. Different metabolic styles they have. Okay, what else? These are cosmopolitan. That means you can find them everywhere. Okay, so based on the type of the structure of the that means the shape of the bacterium, that could be a cocci. Okay, they could be cocci, bacilli. Okay, so they could be cocci. If they are in rod shape, those are called as bacilli, and if they are comma shape, those are called as vibrio. If they are they are forming aggregates, and they are they are coupled in case of diplococci, or maybe they are forming different kinds of shapes by binding with each other, each of such kind of bacteria. Then different shapes may form, and those kind of aggregates are again differently named, kind of streptococci. If they form all cocci together forming a chain shape, then it is streptococci. All these names are very important. Okay, here is the streptococci, and here is the spirillum. Okay, spring shape. So they include 2 major groups. Okay. Here we can see the two major groups. One is Archaebacteria, which is ancient bacteria, earliest residents of the earth. So those are 3.6 billion years ago, primitive organisms. And extreme habitats, they generally resides in extreme habitats, that may be a high temperature or very low temperature, maybe methane concentration, okay, and high salt concentration, like that. So these are divided into three groups which produce methane, or called as methanogens, which allows the high salt concentrations. Those are called as halophiles, which likes the which can only survive in high temperatures, are considered as thermoacidophiles. Okay, these are the classes present in the Archaebacteria. So under eubacteria, eubacteria means true bacteria. So these are again prokaryotes, of course. And what else? They don't have nuclear membrane. Okay, nuclear membrane is absent, and cell organelles are absent. And generally, by hearing the name, we generally come across bacteria means those are harmful, they cause diseases and all. But here are some bacteria, they are useful also. Okay, basically bacteria. So basically, mm, bacteria, they are also involved in the antibiotic production, nutrient recycling, decomposition. Okay, they also present as normal flora. Normal flora means the organisms which present inside the body of other animal, but they won't cause any harm. Instead, they they help the organism to do some to perform some metabolic activities. Those are called as normal flora of that organism. So these bacteria are also present as the normal flora of different organisms. Okay, the cell wall of bacteria is made up of peptidoglycan and teichoic acid. So again, the bacteria is divided into, here is the Monera classification. Monera is divided into Archaebacteria and Eubacteria. And Archaebacteria, Methanobacteria, Halophiles, Thermophiles. Under Cyanobacteria, you can see blue-green algae. So cyanobacteria were also called as blue-green algae. These are not actually algae. Okay, only the name is blue-green algae, but these are not true algae. Because of their photosynthesis habit, they were considered as an algae. So these are also appeared on Earth around 3.2 billion years ago. So before the appearance of these cyanobacteria, Earth atmosphere used to be anaerobic, completely anaerobic. They changed anaerobic habit of the Earth to aerobic. Okay, right. So they are photosynthetic autotrophs. These are photosynthetic. That means they have photosynthetic pigment. So they have chlorophyll. Photosynthetic pigment is nothing but chlorophyll here. And unicellular, colonial or filamentous. So they may be unicellular, or maybe they form some colonies. Okay, by coming together, they may form something called colonies. They may also be filamentous. So tube-like structures, they also have. So many kinds. Okay, the colonies are surrounded by gelatinous sheath. Okay, these kind of colonies are generally surrounded by some gelatinous sheath. Okay, uh, they form blooms. Okay, that means on the water body they form a layer of them. Okay, this kind of bacteria, which generally forms colonies, forms the blooms. That means completely they covers the entire water body like a top layer. That is what the bloom is. So they form the blooms in the water bodies, and they can also fix the nitrogen. That is also a beauty. Fix nitrogen, important thing to remember. They can fix nitrogen, they can perform photosynthetic they can perform photosynthesis, and these are the one who turned anaerobic atmosphere of earth to the aerobic atmosphere. So these are the things. Example is nostoc and anabina. Okay, so these two are the cyanobacteria that can perform nitrogen fixation. So the specialized cells, in the specialized cells, they fix nitrogen, and those are called heterocyst. Okay. So heterocyst is the, here you can see. So here is the heterocyst. Okay. Specialized cell in which the nitrogen fixation will be happened. So coming to the next class, that is bacteria. These are microscopic unicellular. They vary in shapes. Their cells are surrounded by capsule, okay, or slimy layer. What is the difference between capsule and slimy layer? Let's go in detail of the bacterial structure. Okay, a little bit detail we must understand. Okay, generally bacteria is surrounded by cell wall. Okay, one cell wall. Cell wall. If there is any, mostly some outer coverings will also be there external to these cell walls, and those are called as, all together they called as Glyco Calyx. So these Glyco Calyx could be slimy or very tough. Okay, maybe slippery, kind of semi-fluid, that could be slimy, or very tough, that is capsule. So it could be a slimy layer or capsule if it is tough. Okay, then this is called as capsule. If that is slippery or slimy, then it is called as slimy layer. So these are the glycocalyx, that means outer coverings external to the cell wall. Okay, those are present in some bacteria. And what is the component of cell wall in bacteria? That is important. That is peptidoglycan. So peptidoglycan is a major component of the bacterial cell wall. Okay, the genetic material is single covalently closed circular DNA. Okay, this is called a single DNA, and this is covalently closed circular DNA, and some or few plasmids will also be there in some bacteria. Okay, plasmids. So this is the simple structure, and some of the bacteria may have flagella, which is used in the locomotion. Okay, the flagella, the main function of flagella is the locomotion. Okay, and some small projections, hair-like projections called villi, and the singular for the villi is pilus or pilus. Okay, singular term is phylus, and plural is pili. So, and these are also called fimbria. Okay, fimbria. So that's it. Okay. The mode of nutrition. Coming to the mode of nutrition. What is the mode of nutrition? Bacteria generally are having variety of nutrition, kind of, though some bacteria can be autotrophic. Okay. Photoautotrophs. Okay. Some are chemoautotrophs. Among the autotrophs, there are again two varieties, photoautotrophs and chemoautotrophs. And among heterotrophs, they might be a parasite, heterotrophic nutrition, that may be saprophyte, okay, maybe a parasite, or maybe a symbiont. So basically, so here under photoautotroph, what is photoautotroph? That means it utilizes the sunlight. Okay, photoautotrophs, name indicates they can use sunlight to fix the carbon dioxide, similar to the plants. Those are the examples are purple bacteria, okay, green sulfur bacteria, and chemoautotroph. What is chemoautotroph? Chemoautotrophs, actually they derive an energy not from the sunlight. Okay, they won't utilize sunlight. Instead, they use chemical. Okay, chemical energy. So chemical reactions. From chemical reaction, they'll be getting energy. So electrons, they'll be getting electron acceptors and all. So that is called as chemoautotrophic type of mode of nutrition. And what is heterotroph? Heterotroph is the organism which depends on the others for the nutrient requirements. That could be saprophyte, parasite, or symbiont. What is saprophyte? Saprophyte is the organism which generally engulf or abstract food from the dead or decaying material, and parasite is which causes infection to the other host, and symbiont is something which lives on the others with a mutual benefit. Okay, so it doesn't cause harm to the organism in which it lives. Instead, it is benefit for the host organism in which the bacteria is living. Example is the Rhizobium. Okay, example, the best example for this is Rhizobium. What is Rhizobium? Rhizobium is the bacteria which fixes nitrogen in the root nodules of the plants, like pea plants. Okay, example is pea plant. In pea plants, root nodules will be there, and on these root nodules, these Rhizobium bacteria will be colonized. So in that nodules, you can find the colonies of the bacteria. Okay, if you see the anatomical structure, you can find number of bacteria in the root nodule, in a single root nodule, and thereby they fix nitrogen, atmospheric nitrogen to the plant. So that is going to be beneficial for the bacteria, hence it is called as symbiont. So next one is the reproduction. How they will reproduce? Generally, reproduction of the bacteria. So reproduction may be by binary fission. So the bacteria will be divided into two halves, sharing its genetic material as well, and thereby it reproduce. And what else? Even asexual reproduction is there. So this is one of the asexual mode of reproduction. And here, basically, spore will also be formed only during unfavorable conditions. So during unfavorable conditions, spore formation will be formed. So some are called as endospores, which survive in harsh conditions. Okay, these spores are able to survive in the harsh conditions. So the bacteria under favorable conditions, again when the favorable conditions will come back, the bacteria is able to survive. So during harsh condition, these spore will survive. From that spore, the bacteria will be appeared during the onset of favorable conditions. So that is about the spore formation. That is the importance of spore formation. And they also have the sexual reproduction called conjugation. So what is the sexual reproduction? Here they exchange their plasmids. Okay, so that kind of sexual reproduction called conjugation, where the DNA will be exchanged from one bacteria to the other. So using sex pili, they form bridge-like structure between two bacteria. Here one will go to the other. Okay, that kind of DNA exchange will be takes place from here to there. The genetic material is being transferred. So this is called as conjugation, and that is the only mode of sexual reproduction. So the other thing about bacteria, regarding bacteria, is the how the bacteria is classified. Okay, the other way of classification of bacteria is staining process. What is staining? That means adding dye. So there is some important process called gram staining. So in the gram staining process, which is developed by Christian Gram. Sorry, here it should be Gram. So Christian Gram in the year of 1884. So this staining process is able to divide bacteria into two classes. Okay, gram positive bacteria and gram negative bacteria. So here, what is the differentiating factor? The dye, crystal violet. So basically, gram positive bacteria have the ability to retain the primary stain or the crystal violet. So basically, gram negative bacteria, which cannot retain the crystal violet stain, is considered as gram negative. So what happened when generally gram positive bacteria gram positive bacteria will have thick layer of peptidoglycan. Cell wall is very thick, and gram negative bacteria is very thin peptidoglycan. And one more thing is, here in the gram negative bacteria, the cell wall is made up of thin layer of peptidoglycan, but there is external layers which are made up of highly rich lipid content. That means lipid content is very high. So generally the stain will not retain after one alcohol wash. Generally during the process of gram staining, what we do is, first we apply gram stain, that is crystal violet. when crystal violet is applied, both will get the stain, but after an alcohol wash, so alcohol will be applied on the next step, and there the gram negative bacteria, due to thin layer of peptidoglycan, that will be just washed off from the bacteria. But in the gram positive bacteria, the alcohol is not able to completely wash off the gram stain, so it retains. So this will be in violet color. So here is the image showing gram positive bacteria, which appears in the violet color under microscope after gram staining, and this is the gram negative bacteria, which would be in red color, which is generally applied after the wash, alcohol wash. Generally the red dye safranin will be applied. So as the first dye is go off from the gram negative bacteria, the second stain or counter stain will be retained, so that will be appeared in red color. So that is what the gram staining is. So next comes the what else, mycoplasma. So that is another kind of bacteria, mycoplasma. So mycoplasma is previously known as PPLO. Okay. So that means pleuropneumonia like organisms. These doesn't have cell wall at all. So the cell wall is absent in mycoplasma. These are very small, very small organisms. So because of the absence of cell wall, these are resistant to the antibiotics that will trigger the cell wall. That means generally some antibiotics are there which can kills the bacteria which have cell walls. So because of the absence of cell wall, these are resistant towards the antibiotics, which can affect the cell wall. So that kind of for that kind of antibiotics, mycoplasma are resistant. So this is my mycoplasma. So these mycoplasma can live without oxygen, and these are saprophytic, as we already discussed what is saprophytic and what is parasitic. So that is all about the bacterial bacteria. So kingdom Monera is done, and next the kingdom is kingdom Protista. Protista, as we already discussed, these are single celled. Okay, these are single celled eukaryotes. Okay, and microscopic organisms. So they form connecting link to the others. So they maybe form this kingdom Protista, kingdom Protista may form connecting links. That means these are a bit related to the bacteria. These are also related to the higher organisms. Okay, these are having connecting links with the plant animal kingdoms. Okay, that means some of the characters matches with the plant and animal kingdoms. So that is what the connecting link is. So they form connecting link to the others. These are primarily aquatic. All protists are aquatic. Okay, primarily. And they contain, as they are eukaryotes, they contain nucleus, they contain membrane bound organisms like mitochondria, endoplasmic reticulum, and plastids. Okay, they have flagella. Only some of them are having flagella. So dinoflagellate example. Okay, dinoflagellate, two flagella are present. So these are the examples of flagellated protistants. So some are containing cilia. Okay, these are not very well positioned in the biological classification. So basically the boundaries are not well defined. That means some of the organisms from the protesters are considered to be plants or animals by some scientists. Okay, that is what it is. So some of the organisms from kingdom Protista are generally, as they are connecting links, they also have similar characters with the plants and animals. So Some scientists also consider them as plants or animals. So here they form links with the kingdoms like kingdom Plantae, kingdom Animalia, and fungi. They vary in their nutrition. Okay, so they may be, the mode of nutrition may be different kinds. So based on the mode of nutrition, they could be autotrophs, again autotrophs, okay, saprophytes and heterophytes, all are there. So some are autotrophs, some are saprophyte, and some are heterotrophs. So basically photoautotrophs. So basically these are the classes present in the, these are the groups present in the kingdom Protista: Chrysophytes, Dinoflagellates, Euglenoids, slime modes, and Protozoans. Okay, so let us discuss each and every group in detail. Okay, so here are the Chrysophytes. Chrysophytes. So basically Chrysophytes. So the popular example is diatoms. Diatoms. So basically these are very small microscopic marine living floating organisms, and generally they float passively. That means they may not be move themselves, but they have the they will move along with the water current. Okay, those are marine habitants. Okay, they flow through the water currents. Okay, passively they move. These are microscopic, and these are chief producers of the plants. What do you mean by producer? So producer is an organism which produces. That means these are photosynthetic organisms. They can prepare their own food material, and the other organisms generally depends on them. Okay, these are producers of the ocean. So generally these diatoms, as they are bulk in number, so they produce oxygen, they release oxygen. So these are the main producers on the ocean. So this is important point. Okay, so in diatoms, cell walls form two overlapping shells. So they fix together. So one shell and the other shell. So like a soap box. Okay. Soap box. Two shells are stick together in the case of diatoms. Okay. Two overlapping shells, and walls are enclosed with cilia. So here you can find the cilia. Okay, cilia, enclosed with cilia. And cell wall is made up of silica. So here it is also an important point. Their cell walls, diatoms, are made up of the cell walls which are two shells which are having overlapping walls against one another, and those are enclosed with cilia. Okay, cilia, and the cell wall material is silica. So here, due to the wall material is silica, that is not actually soluble in water. So as the diatoms will die, they pile up their wall material, and the process is known as, they form huge bodies which are bottom of the water reservoirs or oceans. Big heaps kind of things will formed. Okay, these are called diatomaceous earth. sorry. So diatomaceous earth. So the process is known as diatomaceous earth. So what else? So these are called as chrysophytes. So because these diatomaceous earth, which is made up of silica, so this is economically beneficial to the human. So beneficial. Okay, so these are used in this grit-like structures will be found. So basically, this soil become gritty and it is used for polishing and filtration of oils. And syrups, oils and syrups. So likewise it is used. So next comes the dinoflagellates. What are dinoflagellates? As name indicates, di means two, flagella means two flagella. So two flagella are present. Mostly these are photosynthetic marine. Okay, marine photosynthetic. Okay, photosynthetic. They appears in yellow, green, brown, blue, or red due to change in proportion of the pigment. So what is the what makes them to change their colors often? That is, they could be any of these colors because of the proportion of the pigments present in their body. Okay, they may be appears in variety of colors, and the cell wall is having thick cellulose plates on outer surface. Okay, see here, this is the dinoflagellate. Okay, these plates, cell wall, so these are cell wall plates. So thick plates, thick cellulose plates on the outer surface. So you can see the small little plates, and most of them has two flagella. One lies longitudinally, so here is the longitudinal flagella. Okay, here is the longitudinal flagella and here is the vertical flagella. Two flagella are there. One is vertical and the other is longitudinal. So one is vertical and one is longitudinal. So it present, this vertical flagella is present between two walls. Okay, between the furrow. So here is a something like groove, and in the inside the groove the vertical flagella is fixed. Okay, between wall plates. So red dinoflagellate, flagellates like Gonialax is the example. Okay, example of red flagella, red dinoflagellate, and repeatedly multiply and makes a sea appears red in color. So wherever the these kind of red dinoflagella is bulk in number, or this Gonialax is present in abundant, then the sea water appears in red color. Okay, generally the toxins released by such large number of large number of these dinoflagellates are able to kill marine animals such as fishes. Okay, they also release toxins. So these toxins, as they are huge in number, okay, they can kill marine organisms like fish. So they show bioluminescence. So this is also a important point. Bioluminescence. What do you mean by bioluminescence? Luminescence. So bioluminescence is the character to produce light. So how they produce light? So because of the oxidative reactions that are happening in their body, they can produce light. Okay. Due to the presence of photogenic granules, some granules are there in their cytoplasm. Okay. And this is popularly known as night light. Okay. Some species of dinoflagellates are taken as food by mussels. So these are preys for mussels. So these are the points. These dinoflagellates are basically having two flagella, and these are marine organisms, also photosynthetic organisms, and they may produce uh, toxins, and they are able to produce light, and those are popularly known as night light. That light is popularly known as night light. So that is what the dinoflagellate is. So here, as we discussed, chrysophytes and dinoflagellates both are photosynthetic organisms. So next comes the euglena. So euglenoid. Euglenoids as euglenoids also, these are freshwater. So earlier what we discussed, all two groups are marine organisms, aquatic organisms, marine, and these euglenoids are fresh water organisms. That means they found in stagnant water, wherever the water stocks, and that kind of fresh water, euglena is present. Instead of cell wall, they have pellicle. Okay. So protein rich layer, that is called pellicle. So instead of cell wall, euglenoids may possess this pellicle, which is protein rich in nature. And because of the pellicle around them, these are flexible. So the cell wall is not tough. Instead, it is made up of a protein layer which is flexible. Okay, that is called pellicle, and two flagella are present. Okay, similar to dinoflagellates, these euglenoids also have the two flagella. One is long flagella and the other is short. So they also perform photosynthesis, and the difference is these are also photosynthetic. Okay, euglenoids are generally photosynthetic. One moment. Okay, so these are also photosynthetic, but if the light is absent, okay, so these are photosynthetic in the presence of light. So if the light is absent, then these are heterotrophic by predating. So these are predators. They eat some other protistans. Okay. The pigments present, as these are photosynthetic organisms, the pigments are present. Okay, photosynthetic pigments are present, which are similar to the higher plants. Okay, that is about the euglenoids. So here these chrysophytes, dinoflagellates, and euglenoids, these three classes are comes under photosynthetic or autotrophs. Autotrophs. Coming to the non-photosynthetic heterotrophs. So what are non-photosynthetic heterotrophic protistans? So here they mostly present as floating organisms that depends on the other protista for nutrition. So all these are basically planktons, plant-like organisms which float on the water, and they are producers, planktons, phytoplanktons. Okay, I'll write it again. Okay, phytoplanktons. So the other organisms which are non-photosynthetic and heterotrophs, those are called zooplanktons. The other groups of protistars are called as zooplanktons. Generally, zooplanktons are dependent on the phytoplankton for their nutrition. Even phytoplanktons or maybe other zooplanktons. These are heterotrophs. Basically, they mostly present as floating organisms that depends on the other protista for their nutrition. So mostly possess holozoic mode of nutrition. That means ingestion. Ingestion is nothing but engulfing the material. Okay, engulfing the solid or liquid material is called as ingestion. So the ingestion mode of nutrition will be performed by these non-photosynthetic heterotrophs. So here parasites and some protistans infect on others. So here some are parasites. Autotrophs are absent in this. Okay, some are parasites and some are symbionts. So parasites, some protestant infect the others which are called as parasites, and some are symbiotes. They live on gut of animals like termites and wood-eating cockroaches, and where they digest cellulose. So symbionts are the organisms which lives on the other's body, but they will not cause infection. Instead, they will be helpful to the organism in which they live. So that kind of symbiotes. Examples are the protistans that lives in the termites and wood-eating cockroaches. Okay, where they generally digest the cellulose. Okay, so reproduction. Generally, reproduction is carried by binary fission, and sexually by zygote formation. So asexually by binary fission and sexually by zygote formation. That means fusion of gametes. So fusion of gametes will be resulted into zygote formation in the case of sexual reproduction, and binary fission is also present in these non-photosynthetic heterotrophs of the protistans. So here some of them are producers and some of them are consumers and some are decomposers. Okay, and here are the examples: amoeba, paramecium, plasmodium, etc. These are, and they could be saprophytes also. So some of them are saprophytes. Okay, heterotrophs. So basically they may be saprophyte or heterotroph. So coming to the next one, that is lime mold. So we come across chrysophyte. Okay, one is chrysophyte, second one is dinoflagellate. Okay, third one is euglenoid, and the next one is slime mold. So all these are autotrophs. So here slime mold is heterotroph, that is kind of saprophyte. So example is Fusarium. Their body move along the dead and decaying twigs and leaves engulfing the organic material. So they move along the dead body and engulf the organic material that is decaying. Okay, they form aggregates called plasmodium. So the aggregates are called as plasmodium. Is the aggregate. Aggregate called plasmodium. And during unfavorable conditions, the plasmodium differentiate. So these kind of plasmodium will differentiate and forms the fruiting body. So these kind of fruiting bodies will be formed on the plasmodium. Okay, these kind of fruiting bodies will be formed which bear the spores on the tips. So these are spores. They can spread and differentiate into new protestant or the slime mold. So because of the presence of slimy layer around these plasmodium, those are called slime molds. Okay. During unfavorable condition, the plasmodium differentiate and forms the fruiting bodies which bears these spores at their tips. And spores have strong resistant cell wall. So these spores are having resistant cell walls. Okay, very tough cell walls. So they can survive through the harsh conditions. Okay, adverse conditions. The spores are dispersed by air currents. So these kind of spores will be dispersed by air currents. Okay, and example is Dictyostelium is a kind of slime mold. Okay, here is the image showing plasmodial strand. If you go for the cross section, you can see this kind of ectoplasm in which, okay, all cells are accumulated and endoplasm will be there. Okay, actin bundles, that means the cell cytoskeleton structures. These cytoskeleton will be formed like this, and here you can see the spore formation. So they will be cut, they will develop and cut into the new things. Okay, spores will be appeared on top at tips. So next one is the protozoan. Okay, these are completely, as we discussed, slime modes are saprophytic, and here is the heterotrophs, protozoans, in which amoeba will come, paramecium will come. Okay, all these things will come under protozoans only. So protozoans are the protists which are either lives as predators. Okay, protozoans are predators or parasites. That means they also cause infection. So what is the example? Amoebic dysentery caused by Entamoeba histolytica. So that is the example. So they are considered as primitive relatives of the animals. These are primitive relatives of the animals. And again, there are 4 groups based on the mode of locomotion. How do they move? Locomotion. So here are the amoeboids, which possess pseudopodia. Amoeboids. Okay, if they possess flagella, those are called flagellated protozoans. And some are ciliated protozoans. And some are sporozoans, which doesn't move. Okay, they don't have locomotory organs. Okay, so here are amoeboid protozoans. They lives in fresh water. Okay, example is amoeba, antamoeba. Okay, seawater. Seawater is the example is radiolaria, and moist soil, which have the high moisture content. Okay, some are parasites. which causes amoebic dysentery and amoeba. And they move and capture their food by pseudopodia. Okay, so that hence these are called amoeboid protozoans. They possess pseudopodia. Pseudopodia is the false foot. Sorry. Or pseudopodia. Okay, some are having okay, marine forms have the silica shells on their surface. So marine forms like radiolaria. Okay, this is an amoeboid protozoan only. That means they form pseudopodia, and these kind of forms have silica shells on their surface. And next one is the flagellated protozoan. Flagellated protozoans are basically free living or parasitic. Okay, either free living or parasitic. So because of the presence of flagella, they can move and they cause diseases like sleeping sickness, that is caused by trypanosoma, very important. Okay, trypanosoma causes sleeping sickness. Leishmania donovani causes kala azar or dum-dum fever. So here are the two diseases that we discussed, that is sleeping sickness and kala azar, or the dum-dum fever. So sleeping sickness is caused by trypanosoma. Okay, Leishmania donovani causes the kala azar or the dum-dum fever, and sandfly is the vehicle for kala azar fever. So that means it transmits the parasite. Okay, this Leishmania parasite will be transmitted by sandfly. So here the other examples are Giardia intestinalis. Giardia intestinalis causes giardiasis disease. Okay, Trypanosoma cruzi causes the South American trypanosomiasis. Okay, the Changas disease. So all these are important. Okay, any other disease may come and asking the causative agent or like that, any type of questions will come in this. So next one is the ciliated protozoans. Ciliated protozoans, these are aquatic, actually moving organisms. Okay, aquatic organisms, actively moving organisms due to the presence of number of cilia. So maybe all over body the cilia may be present. Okay, these cilia they helps to move, and also these cilia sorry, helps to move the food. So if there is some food source, then cilia, okay, will show flip kind of movements. So because of this because of these ciliated movements, cilia movements, the food will go towards the oral cavity. Okay, or gullet. So oral cavity is called as gullet, and food will move towards its oral cavity, that is gullet. Example is Paramecium. So they have two nuclei. One is big nuclei called vegetative nucleus. So here I think we have Paramecium image. So you can see here. Okay. So in the Paramecium structure, sorry. You can see meganucleus and small, that is micronucleus. Okay, so basically meganucleus is also called as vegetative nucleus. It doesn't have function in the reproduction. And one small nucleus, micronucleus, where are we? So these kind of two nucleus are present. So the small nucleus is also called as reproductive nucleus because it is involved in the reproduction. So next one next group is Sporozoans. So they doesn't have locomotory organs at all. So here are the three classes. So amoeboids which have the pseudopodial structures, and flagellated protozoans which consist of flagella. Ciliated protozoans consist of cilia, but sporozoans doesn't have locomotory organs. So they include diverse organisms that have infectious spore-like stage. Okay, in their life cycle. In their life cycle, they have spore forming stage, which is infectious. So that kind of organisms are comes under sporozoans. So what is the example? So this is here is the plasmodium. Plasmodium, which causes malaria, is the popular example. Popular protozoan which comes under sporozoan. So here are the 4 protozoans. Next comes the kingdom fungi. So as we discussed, we completed 5. Sorry. So we completed, yeah, here you can see 5 groups of, so 5 groups of protistans. So protista, these are unicellular organisms. Okay, it's very small microscopic organisms only. So based on the nutritions, we can divide them into different groups. Some are autotrophic, saprophytic, and heterotrophic. Among heterotrophics, trophs, we can also have parasites and free living organisms. So next comes the kingdom fungi. So this is all about the kingdom protista. Next comes the kingdom fungi. So all fungi comes under kingdom fungi. So it consists of unique kingdom of heterotrophic organisms. So it is completely consist of only heterotrophs. Only heterotrophs are present. Heterotrophs, no autotrophs at all. Okay, which show great morphological and habitual diversity. So they may be very small. Okay, they can live on very variety of habitats. Okay, examples are mushrooms, toadstool, etc. And these are cosmopolitan. Wherever you go, you can find these fungi. Okay, and you can they may be present in soil. Okay, air, maybe in other animals or plants, in other organisms, maybe as parasites, maybe as maybe as symbiotes. Okay, example is lichen. Okay, parasite Puccinia that cause the disease in animals, plants. So Puccinia, Penicillium. Okay, and some are used in so apart from the diseases they cause, they are also useful to the human being in some way. So what are the benefits? They generally involved in fermentation. So example is yeast. Yeast is used in bakery industry for making bread and all. Okay. So generally fungi are filamentous, tube-like. Okay. And one exception, that is unicellular simple fungi, that is yeast. Yeast is not filamentous like this yeast is, and the remaining are filamentous. Okay, their body consists of thin, slender, thread-like structure called hypha. Very thin, thread-like structures. called hypha, and these hypha entangle to give rise to the structure called mycelium. Okay. So generally, their body consists of thin, slender, thread-like structures called hypha that makes a network called mycelium. Some hypha are continuous tubes with multinucleated. That means this hypha generally is made up of many cells, but these cells doesn't have cell walls or separations. Okay, septa to separate one cell to the other. All cells together makes a filament, and among in the inside the filament you can see many nuclei. Okay, so this is called cenocytic condition. Okay, here this is called cenocytic condition, and in some fungi, okay, they have cross walls among them. So some fungi have cross walls between one cell to the other. In the filament you can see the cross walls, and those are multi-celled. So continuous tubes with multi-cell nucleated cytoplasm is known as cenocytic condition. So here is the cenocytic condition. That means many nuclei without septa. So most of them are heterotrophic, and they absorb soluble organic matter from dead substrates. Hence, those are also called as saprophytes, and some are parasites, and some are symbionts. Example is mycorrhiza, and this mycorrhiza present in the plant and fixes nitrogen. And this association is not very specific. Okay, a plant can associate with any kind of mycorrhiza, not species specific. Okay, in the plant root system, many kinds of mycorrhiza may be present. So, and one more thing, if they are present outside of it, then it is called as ectomycorrhiza. Okay, if those are present, ectomycorrhiza, and they are present, if they are present outside external, then those are called ectomycorrhiza, and they are present internal, then those are called as endomycorrhiza. Okay, internal to the organism. Okay, ectomycorrhiza and endomycorrhiza. So the mode of nutrition, mode of reproduction, so that may be vegetative for fungi. Vegetative reproduction is there, asexual reproduction is there, and sexual reproduction is also there. So vegetative reproduction is done by fragmentation, fission, and budding. Okay, that means the fungal hypha may be fragmented, and each fragment may be developed into a new fungi, and maybe fission, and maybe budding. Small buds may appear and each bud may develop. So asexual reproduction by spore formation, and spores called conidia or sporangiospores or zoospores. Okay, these are the asexual spores. So conidia, ascospores, and Basidio spores. Zoospores are these are the asexual spores. And the other type of spores are sexual spores. Those are oospores, ascospores, and Basidiospores. Okay. So these three are the sexual spores. Spores produced in specialized structures called fruiting bodies. So generally, something called fruiting bodies will appear on the filaments. Okay, inside which the spores will appear. These are fruiting bodies. Bears the spores in them. So this will rupture and spores will enter into the atmosphere, hence by spread. So basically, two haploid hypha compatible during sexual reproduction may be compatible hypha, two hypha may be come closure like this, and they fuse and exchange. Okay, they come closer and fuse. In some fungi, fusion of haploid cells immediately result into the diploid cells, and in some other like ascomycetes, what will happen? The intervening dikaryotic stage is present. That means here these two compatible hypha will be fused together, and so these kind of fertilization will be happen in two nucleus. They generally after the protoplasm is fused, okay, cytoplasm is fused to give rise to a single cell. The nucleus may also be fused to form diploid cell, but in some cases this diploid stage will not appear immediately, but intervening dikaryotic stage is present. Later, the parental nuclei will fuse and become diploid. So for some time, this dikaryon will be present. Dikaryotic stage is present, and then the nucleus will fuse to produce the diploid. So the fungi produces fruiting bodies, and reduction cell division occur, leading to the formation of haploid spores. So from here, diplotic stage, the meiosis will happen, and thereby forming the spores. So basically, based on the morphology of the mycelium, generally this is the mode of reproduction. So these fungi, the kingdom fungi is divided into phycomycetes, ascomycetes, okay, Basidiomyces and Deuteromycetes. So these are the four classes. So here, kingdom fungi, is divided into, okay, ascomycetes, okay, Basidiomycetes, sorry, I forgot to write phycomycetes. So here are the phycomycetes, ascomycetes, Basidiomycetes, and Deuteromycetes. So what is the difference? What are the bases? Based on the hyphal structure. So under phycomycetes basically found in aquatic habitats, okay, decaying wood and moist places. So these are obligate parasites. In plants, so these are obligate parasites. That means they only survive through the in the other bodies, plant bodies only. Okay, so they can only survive by parasitism. Okay, by infecting the others. So they are mostly present in aquatic habitats, and maybe some other kind of moist places. So asexual reproduction is by zoospores or aplanospores. Okay, these zoospores are motile, and aplanospores are non-motile. Okay, during sexual reproduction, so they perform asexual reproduction by producing zoospores and aplanospores. Sexual reproduction by by zygospore formation. Okay, zygospores will be formed. So that means fusion of gametes will be there, and if the gametes are asymmetric, that means they are similar in morphology, those are called as isogamous. And if they are not similar, then one is large and other is small, like that, they are anisogamous. So these all about the sexual reproduction. So zoospores, aplonospores comes under asexual spores. Zygospores are appeared in the sexual reproduction, and that might be isogamy or anisogamy. Okay. So some common examples are leucar, okay, leucar, rhizopus, that is bread mold, and algo, that is parasitic fungi on mustard plant. So the other is ascomycetes. So ascomycetes, these are sac fungi. So sac fungi, mostly these are multicellular. Example is penicillium, rarely unicellular. So under sac fungi called ascomycetes, yeast will come, yeast and penicillium. So these are the examples. So basically, mycelium is here branched and septate. So you can see branches like this. So branched and septate. So in ascospores, mycelium is branched and septate. Some are unicellular and some are multicellular. Okay. And these are reproduced through asexual reproduction by producing conidia. So fruiting bodies are called as conidia, in which conidiospores are found, and sexual spores are called as ascospores. So all these terms are important. Conidia or conidiospores are produced in conidia, in ascomycetes, and zoospores, aplonospores, zygospores are comes under phycomycetes. Okay. These ascospores produced in a sac. That's why these are called as sac fungi. Okay, these ascospores are produced in sac-like structure called asci. Okay, singular is ascus here, and the fruiting bodies are called as ascocarps. Okay, ascocarps. So basically examples are Aspergillus, Claviceps, okay, Neurospora. Neurospora used extensively in biochemical and genetic work, and many members like morels and truffles are edible and considered as delicious. So some edible fungi is also there. Okay, here Neurospora, molecular studies. Okay, and what else? Branched and septate. Their mycelium is branched and septate. These are called as sac fungi because of the presence of asci. Okay, the sac is called as asci. Okay, so basically sac-like structure called asci. In the asci, ascospores are present. Okay, fruiting bodies are called as ascocarps. Some fungi are edible. Okay, and considered as delicious. Those are examples are morels and truffles. So Basidiomyces. Next one is Basidiomyces. So what are Basidiomyces? Basidiomyces are club fungi. Okay, these are club fungi. So ascomycetes are sac fungi. Okay, so phycomycetes are basically lower fungi. So here basidiomycetes are club fungi, and these are commonly known as basidiomycetes or mushroom, common forms of mushrooms. Okay, example is mushroom, and breakfast fungi are puff balls. So all these are things. So basically, here is also the mycelium is branched and septate. Sorry. Branched and septate. Okay. The septum consists of barrel shaped. So barrel shaped pore. So and here all these are having septa. Okay, these septum consist of a pore which is having barrel in shape. So all septum consist of barrel shaped pores, and these are called as, okay, these branched and septate filament is there, and each septa have the barrel shaped pore, and these type of septum which is consisting of barrel shaped pore is known as dolipore system. Dolipore. So dolipore septum. So this kind of septum is known as dolipore septum. So here asexual spores are generally not found. So in Basidiomycetes, no asexual spores. Asexual spores are absent. Okay, this is one of the important features. So we need to remember which spore is happening in what, which group. So if it is zoospore or zoospore, then aplanospore, then it is phycomycete. Okay, if it is ascospore, okay, aplanospore is belongs to phycomycetes, and aplospore is belongs to ascomycetes. So basidiospores doesn't have the spores at all. Okay, basidiomycetes doesn't have the asexual spores. This vegetative reproduction is present in the form of fragmentation. Okay, the sex organs are also absent. No sexual reproduction. So only vegetative reproduction in the form of fragmentation. But how the genetic material is exchanged? Through plasmogamy. So plasmogamy is the case where vegetative reproduction, the fusion of two vegetative hypha will happen. So in this vegetative hypha of two types of basidiomycetes will fuse together. Okay. Somatic cells of different strains or different genotypes of the mycelium will fuse together, and resultant structure is dikaryote. Okay, dikaryote. That means two nuclei are there, and which ultimately gave rise to the basidium. So that is what the basidiomycete, the word comes from that. Okay. Basidium, the karyogamy and meiosis will take place. So here in these two nucleus, they fuse together to give zygote, in which the that is what karyogamy is. 4 gametes will form, resultant of meiosis. Okay, these are called Basidio spores. Okay, these Basidio spores. So this is what the Basidium is. So this is called Basidium. Inside where Basidio spores are happening, are producing. Inside Basidium, okay, Basidio spores are present, and inside these Basidium are arranged in the fruiting body called Basidiocarp. So basidiocarp, basidium inside basidio spore. So this is basidium, and these kind of basidiums are present inside the basidiocarp, and this basidium will produce basidiospore. So examples are agaricus and Eustyleva and deuteromycetes. Next one is so deuteromycetes. These are imperfect fungi. Okay, imperfect. Why these are imperfect fungi? These are not classified perfectly because the only asexual or vegetative phases of these fungi are known. Okay, when sexual reproduction is not known. So if their sexual reproduction is how they are producing sexually, if that is the participants in the deuteromycetes, if they are studied well, then they may be put into the some of these groups, maybe phycomycetes, maybe basidiomycetes, or maybe in other class ascomycetes. So like that. These are not well classified because of the because these are not well studied for their sexual forms. When the sexual forms of these fungi were discovered, they moved into the classes they rightly belongs to. So deuteromycetes reproduce only by asexual spores known as conidia. So asexual reproduction is by conidia. We also find another conidia somewhere in the where. So conidiophores and conidia are also present in the ascomycetes. Okay, sorry. So here is also conidiophores in the deuteromycetes. Okay, and their mycelium is also branched septate. So we have branched septate mycelium in the ascomycetes, basidiomycetes, and deuteromycetes. These three groups also have branched and septate. Okay, conidia is present in ascomycetes and these deuteromycetes. Okay, so mycelium is septate and branched. Some members are saprophytes, parasites, while large of them are decomposers. Decomposers of litter helps the mineral recycling. So if those are decomposing the litter, they helps in the mineral recycling. Some examples are Alternaria, Coletrotrichum, and Trichoderma. This is all about the kingdom fungi. Okay. So today we discussed we were continuing with the Monera, kingdom Monera, in which we covered eubacteria, which is divided into cyanobacteria and bacteria, 2 classes, and kingdom Protista, which have the 5 groups: Chrysophytes, Dinoflagellates, okay, euglenoids, and slime molds, and the protozoans. So likewise, the fungi, the kingdom fungi, we also discussed the kingdom fungi. Okay, what are the reproductive forms of kingdom fungi, and what are the mode of nutrition? Okay, what is the mycelium structure of mycelium, and all these things. Okay, there are 4 types: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Phycomycetes are a bit primitive or lower fungi. Okay, and here zoospores will be formed during their asexual reproduction. Zoospores will be formed. Maybe zygospores, zoospores, aplonospores are considered as asexual spores. Okay, haplonospores are considered as the sexual spores, and under ascomycetes you can find the conidia and ascospores. So the asexual spores are ascospores. Okay, produced in the body, fruiting body called asci. Okay, the singular is ascus. And Basidio spores, which is called as club fungi. Okay, these are asexual reproduction is absent. Sexual reproduction is also absent in Basidiomyces. The vegetative reproduction only through fragmentation is possible. So how the genetic exchange will be happen? So through plasmogamy, that means fusion of protoplasm. Okay. Deuteromycetes are imperfect fungi. These are the examples. Okay. So here ends the biological classification. We left with kingdom plant and kingdom animalia, which have the separate chapters itself. Okay. So we will discuss kingdom plant in the next class, followed by kingdom animalia. Thereby ends the first chapter called diversity of the life. Is there any questions? Are there any doubts to discuss?
No, ma'am.
No. So are you getting them properly? So tomorrow you're going to have the exam. Okay.
Yes, ma'am.
Okay. On the topics which have covered in this week, you are going to have questions. Okay, we'll again discuss the previous questions from previous exams. Okay, previous
Yes,
exam papers and all in the coming classes. Okay.
Yes, ma'am.
Okay, that's it. Thank you. Bye. Is there anything to discuss now? Okay, then you can leave. Okay. I'll message in the group when the next class going to be, at what time it is going to be. Okay. So
Okay, ma'am.